A very good day to all judges. My name is SANISHA, and today I will be presenting my research titled Network Analysis of COVID-19 Genes for Long COVID. The coronavirus disease 2019, with over 430 million documented cases and over 5 million deaths as of early 2022, has had a massive impact on people's lives worldwide. The virus, SARS-CoV-2, penetrates human cells via the ACE2 receptor in the cell's membrane. Recent clinical evidence has brought to light certain cases in which the symptoms of this disease tend to persist in the infected individual. This condition has been named long COVID, post-acute sequelae of COVID, or post-acute COVID-19 syndrome. As seen in the figure on the right, SARS-CoV-2 has effectively demonstrated that it can bring harm to multiple organs, as long COVID is a multi-organ system disease. So while conducting the literature review, it was found that similar to COVID-19, long COVID can also be categorized into based on the duration of the persisting symptoms in an individual. So the symptoms last beyond 3 weeks, then the individual may have post-acute COVID, and if the symptoms last beyond 12 weeks, then chronic COVID. Some of the risk factors identified by the National Institute of Health and Care Excellence are the female sex, poor pre-pandemic mental health, poor general health, the prevalence of any comorbidities such as diabetes or asthma, and of course the number of symptoms during the acute infection, where the higher number of symptoms in the acute infection, then the higher the chances of a person actually developing long COVID. So hypotheses for the pathophysiology of long COVID include sequelae of organ damage, chronic inflammation, accompanied by dysfunctional stimulation of the autonomic nervous system, and sequelae of critical illness. So due to the early unawareness of the range of symptoms brought about by COVID-19, most of the population, they do not build an immediate connection between their symptoms and the disease. Patients and patient advocacy groups have voiced out about the delayed recognition of long COVID symptoms and how that has led to insufficient support for them and the overwhelmed healthcare systems. Therefore, there is a need to identify specific biomarkers that relate to each symptom to advise the general population, experts in treatment options and the mechanism of long COVID. So the hypothesis of this project is to analyze the multiple symptoms genes with long COVID genes with different bioinformatics tools. Our research objectives, the first one is to recognize common persistent symptoms in patients with long COVID. The second is to identify the genes responsible for long COVID symptoms by gene prioritization and ranking. The third is to identify the overrepresented genes by gene enrichment against SARS-CoV-2 interactome references. And the final one is to obtain FDA-approved drugs for the top genes identified. So as for our methodology, we have searched through all publications reported for long COVID symptoms like systematic reviews, meta-analyses, and other publications from online public repositories, and we've collected more than 250 symptoms of long COVID. Then we created 2 sets of gene lists, one containing the differentially expressed genes for SARS-CoV-2, and another containing the phenotypic gene associations for each of the symptoms that we identified. Then we did the process of gene ranking, which was conducted using these data sets that we constructed in step 1 to identify the common elements. This information tells us which genes in SARS-CoV-2 may possibly influence the onset of certain symptoms. Following gene ranking, Cytoscape was used to identify the hub genes, and the drug gene interaction database was used to find drugs that have a high interaction score with each hub gene. The final step was gene enrichment analysis to get details on which biological pathways each drug plays a role in. So if you look at the results, figure 1 shows the 255 symptoms that we identified in total from different organ systems in the body. Gene lists were downloaded for each particular symptom from the Human Phenotype Ontology database, where we obtained the phenotypic gene associations for each symptom, and then they were classified into 10 categories. Figure 2 shows the hub genes and FDA-approved drugs identified for all the symptoms collectively using Cytoscape and the drug gene interaction database. So in figure 2, we have a network of all the hub genes, and below that we have some of the drugs that we identified that have a high interaction score with these particular hub genes. There could be further modified into possible treatment options. Okay, figures 1 to 4 show our results from the gene enrichment analysis step. We've got KEGG pathway, we've got biological processes, molecular functions, and cellular components. These tables highlight which particular pathways in our body the heart genes are actually involved in. In this way, we know what role exactly they play, so we can further identify how the treatment options can help with the symptoms, help alleviate the symptoms. So the identified long COVID involving 255 symptoms may potentially alter evolutionary conserved second messenger signaling cascades via activation of G protein coupled receptors or GPCRs by directly modulating the signaling. And just to give you an idea of how significant GPCRs are, they're involved in processes that regulate our emotions and behavior, our immune system, our interaction with various proteins and signaling molecules, light perception, even our sleep cycle. So the GPCR's adaptability to changing environmental conditions is based on their ability to bind varied ligands, change conformation, up or down regulate membrane expression, and interact with other proteins and receptors. This is a database, a website that's currently being developed by my co-authors and I. It's called the Long COVID Gene Association Resource. It contains all of our findings from this research, all of the details, gene enrichment analysis results, the gene list for each particular symptom, divided by categories, all available on this website, and it's still currently being updated. So here's the link for anyone interested. In conclusion, long COVID has been established as a major global health concern, but because the disease is still in its early stages, it is difficult to predict its long-term prognosis. The identification of 255 symptoms in the different organ systems, the biomarker discoveries and respective FDA-approved drugs, as well as their associated gene ontology and pathway insights, we aim to aid in determining the etiology of long COVID and then customize treatment by providing healthcare professionals around the world some ease in diagnosing long COVID in their patients. So the authors would like to acknowledge the Department of Diagnostic and Allied Health Sciences, Faculty of Health and Life Sciences and Management Science University, Shah Alam, Malaysia. We'd like to thank them for giving us the opportunity and research facilities to conduct this research. These are my references, and that is all for me. Thank you very much.